I think it's interesting that the culture media is one of the most closely held secrets. When you talk to somebody about a bioprocess, they might tell you all about their downstream process, what resins and what they might tell you about their cell line and things like that. But the culture media is like, oh, no, we can't disclose that. In a way, I guess I take that as a compliment or some kind of encouragement that what we're working on is important. I remember, again, when you go back 30 years, nobody cared about the media. Was just that stuff you bought. I think there's more appreciation for what we do on the culture media development side.
Hey there, biotech innovator. Welcome to another episode on the Smart Biotech Scientist. I'm your host, David Brühlmann, and today we're diving into the fascinating world of cell culture media development with a true industry pioneer. Joining us is Tom Fletcher, Research and Development Director at Fujifilm Irvine Scientific, with over 30 years of experience developing media for large-scale production processes. In part 1 of our conversation, we'll explore the fundamental shifts in biomanufacturing, current challenges in cell culture media development, and how automation, machine learning, and AI are transforming our industry. So grab your coffee, your notebook, and let's get started with today's episode. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Tom, to the Smart Biotech Scientist Podcast. It's so good to have you back.
I'm excited to be back, David. Thank you for inviting me.
Absolutely. It's a pleasure. And I've been looking forward to having you back on. And by the way, the first episode we did a couple of years ago on rational media design is still the most popular, the most downloaded episode in the history of the Smart Biotech Scientist.
Well, I don't know what to say about that, David. I don't know how to explain it, but I'm just pleased.
Absolutely. It's amazing. Since you have been part of this adventure and you have also listened to many episodes, I would like to ask you this question I love to ask at the beginning of my interviews. Share something that you believe about bioprocess development that most people disagree with.
Okay, well, I can't say I've listened to all the podcasts, by the way, but that's a creative question. I had to stop and think, and I think I would turn that question around and say, what do most people believe, or what do many people believe that I disagree with? I mean, it's almost the same question, I realize, but on this subject, we're going to be talking a little bit today about artificial intelligence and algorithms and things like that. And I've honestly heard from people who think that it's all kind of a worthless endeavor because, and this comes from experienced people like in late stage process development and things, and they're skeptical of some of these new tools. And so I disagree with that. I mean, I think there's tremendous promise in some of these new tools. When I listen to them, why do they disagree? I understand to some extent. The complexity of a bioprocess makes it very challenging. And then they would also provide me with evidence of how few case studies there are. There's very few case studies of true success, especially at scale. These are typically late stage people who are saying, don't waste my time. Like at a conference, I'll say, wasn't that an exciting presentation? And I'll say, oh yeah, that stuff. We don't even listen to that stuff. So I disagree with that, okay? So there's another thing though. I have two things that I disagree with that I hear from other people. The other one is actually on the opposite end of the same question, because I hear from people who say, oh yeah, these algorithms, we can just have these in silico algorithms we really don't need to work so hard anymore, because as long as we have the right program, maybe we license it or something and we've got this sophisticated program, now we just need to push the right buttons and we'll get these process development issues resolved so efficiently. And so I disagree with that too. I think they fail to appreciate, well, we'll be talking about this a little bit more, but I think they really fail to appreciate how these tools can enhance process development. So anyhow, I guess that's a great question to start out this topic, because we're going to talk more about that.
Yeah, I'm excited to unpack these points. Now, when we last spoke, Tom, you shared some fascinating insights about your journey in biotech. So for those of the listeners who have not been able to listen to this episode, can you walk us through your career path and tell us a bit these unexpected turns and aha moments that you experienced?
I think I'll leave out the surfing stories this time, but I did, and I don't know how unusual my path is. I studied biological sciences, and that's simply because I was interested. I really never had a career in my mind, and I still kind of don't, honestly. I'm happy that I get to work on things that I enjoy. But I have always admired biochemistry in particular for kind of the elegance, if that's the right word, of compared to other branches of chemistry, the efficiency of biochemistry and the specificity, the apparent design that you can see in how biochemistry works. So I'm fascinated by structure-function relationships. I'm fascinated by things like molecular machines and things like that. So it's always held my interest, and I've just been attracted to the field. Thankfully, I had a chance to work in protein chemistry research when I was still in school, and so I really enjoyed that research. And honestly, when I got involved in what we now call the industry, it was just a job. I thought, well, I've got this background in biochemistry. I started out doing everything from blood fractionation. I got involved at that point in cell culture media manufacturing. And so just one thing led to another. I honestly learned cell culture on the job. I never learned cell culture in school. So I've been in the industry a long time. I remember the days of early serum-free media and how we graduated into animal component-free, and now it's almost all chemically defined. But in terms of what keeps me motivated, I think there's a huge upside potential in this field. I think there's still a lot to be discovered in terms of how to support the kind of results we want to see in a cell culture process. We want to see improvements in, titers, productivity of whatever type we're looking at, product quality, and the culture media, I believe, is a key factor in that. So yeah, I'm still highly motivated to develop new and better cell culture media solutions to the current problems.
With over 30 years in the industry, and more specifically in cell culture media development, what fundamental shifts have you witnessed that has most impact how pharmaceutical companies approach manufacturing today?
Well, if we go back 30 years, again, I kind of went over that evolution from serum-free, which was kind of the talk of the town back in those days. I was a little bit surprised when one of our early customers said, "Thanks for achieving my performance goals. Now I want you to eliminate all the animal components." And we had several things in there. They were pure proteins, serum-derived proteins and things like that. But we thought, wow, how are we ever going to get rid of these things? And we found ways of, for example, using vegetable-derived protein hydrolysates and things. So I've kind of lived through all of those things over those many years. But today, it's almost all chemically defined, and I think that's a huge breakthrough. In fact, as I've watched those categories change, I don't know of anything more advanced than chemically defined. I don't think there's a next category when it comes to being defined, because we're completely defined. We are still learning about the importance of the impurities, for example, or also how to balance these nutrients in ways that achieve the maximum performance, and obviously how to do that in less time, more efficiently. I think it's interesting that the culture media is one of the most closely held secrets when you talk to somebody about a bioprocess. They might tell you all about their downstream process, what resins and what they might tell you about their cell line and things like that. But the culture media is like, oh, no, we can't disclose that. In a way, I guess I take that as a compliment or some kind of encouragement that what we're working on is important. I remember, again, when you go back 30 years, nobody cared about the media. Was just that stuff you bought. I think there's more appreciation for what we do on the culture media development side. In a way, I guess I felt in the beginning there wasn't much respect for culture media development, but today it really is part of the plan. Sorry, I'm giving you several answers to the same question, but I think Nowadays, people will typically start with a platform culture media just for the sake of speed, right? So I think that's become fairly common, and that was not at all common back in those days. So you'll start with a platform, but you'll also plan in your timeline for some optimization maybe in the later stages. So I think those subjects, people have learned with time to do those two things, start with a platform, but eventually also allocate resources so you can optimize at the right time.
Yeah, we have definitely come far away in media development. Now it's chemically fine. Last time we talked about rational media development. There are a lot of strategies to streamline that and to accelerate that. But if we look now at the media development today, what are the challenges we're now facing?
In some ways, they haven't changed much. We still deal with the complexity of culture media formulas. Even in a chemically defined world, we've got 60, 70 or more components in a typical medium. And that's not just about what's required for the process, because now we also, especially as we intensify processes, higher cell densities, higher concentrations, especially in feed media. So we see a lot more challenges in terms of interactions of these components. We see redox reactions, which seems like we should have anticipated that, but now we're sometimes surprised by things that can happen. And the redox reactions can be slow, so this is where we get caught by surprise. We find out, oh, because we mixed it for so long, now we're filtering out some of these important metals, for example, participate in the redox reactions. I mentioned earlier impurities too. We're learning that it's important to control impurities because some of these trace metals like manganese can be critical to achieving the product quality, the glycosylation that we want. So none of these subjects are particularly new, but they're becoming more and more part of what's expected during media development as they're watching out for these interactions. We're learning how to concentrate things, especially feed media. And there are actually now algorithms for helping to bucket the components so that we learn how to prepare the medium. Let's say take one group and dissolve it completely, dissolve another group maybe at a different pH, and then combine them. There's a lot of kind of tricks of the trade. They end up seeming kind of primitive or practical because it's not really, so much about nutrition as just getting this liquid to dissolve and be stable. I mean, that's another thing, actually. We're focusing a lot on ways to stabilize these concentrated media, say for a perfusion process or for a feed. So again, I'm probably giving you a lot of answers to one question, but these are the types of challenges that we face today as we develop media. It's not just achieving the performance, but it's some of these more practical issues.
And do you see a shift in the way we approach these challenges?
Yeah, as we learn, we become better at anticipating the challenges. And one thing probably I should mention, because it's been a big part of my life recently, is we're actually developing automated liquid preparation methods. So we've been working on a single-use technology. I think I mentioned it to you once. I'm still excited about it because it can really change the landscape in terms of media preparation. Again, something nobody thinks about, but once you see how efficient it can be, and I think the key attraction is how reliable. Once you have an automated process, you've taken out this human factor as a variable. Yeah, it's hard for me to describe it with words on this podcast, but I mean, it's just really, I think, a game changer because now the media prep can be so much more reliable.
Yeah, this is a great transition towards the technology sides. I want to focus on a bit more in this episode because when you say media development, well, there are several aspects to it. You mentioned the whole composition, so the chemical side, what is in the media. So now, as you mentioned, there is the automation side or the industrial side. Like, how do you consistently prepare your media batches at a large scale? How does that work using automation and other technologies? And so, obviously, throughout your career, you have witnessed a tremendous evolution of advancement in the industry, going from very manual processes now to increasingly automated processes, now more recently machine learning, AI. We have a lot of different technologies at our disposals today. So here is the $1 million question. How can we as biotech scientists today effectively leverage these technologies while still ensuring they enhance rather than replace human understanding and our expertise in bioprocess development?
Yeah, no, I agree. That is a key question, and it deserves people responsible for this development activities to really think it through, because I think these tools are valuable, as I implied earlier on. I think applying them in the right way is so important. And I think in your question, you kind of implied the answer. We never want to replace the human part of this collaboration, if you will. I recently heard of a lecture, and I think there's been some papers written about how the theme was AI plus HI equals CI. So that's artificial intelligence plus human intelligence equals collaborative intelligence. Now, that's just a bunch of words, but I get what it means, because we don't at all want to minimize the influence of our human experts. We don't want to neglect the human experience and how important it is to generate the knowledge that we're trying to achieve. And I like to point out that the goal, and I do this with our group here at Fujifilm, I like to point out to the R&D group that what we produce in R&D is knowledge. What the business does with that, or what the customers do with that, is not as much. What we need to worry about is generating the knowledge. And the machine learning and the artificial intelligence, it can generate information, it can generate interesting things. But to really learn and to know something, I think the human is a key part of that. For one thing, before we conduct an experiment, I think it's important to look at the experiment as a way of testing a hypothesis. And so I really encourage, again, scientists to think ahead of their experiment about what questions they would like to answer in terms of a hypothesis, so that when we test the multiple factors, we get an answer to a question and we become smarter, more knowledgeable. It helps me anyhow to think that what we're trying to generate is knowledge. And even though a machine can learn, there's a difference between learning and knowledge, or really understanding something. With that in mind, I think people need to strike a balance and learn how to collaborate with these new tools, have the humans collaborate with in silico tools to get the best result.
That wraps up part 1 of our conversation with Tom Fletcher. We've explored the impressive evolution of cell culture media development over 3 decades and gained valuable insights into balancing traditional expertise with cutting-edge technology. Join us for part 2, where we'll dive deeper into AI implementation strategies and Tom's vision for the future of bioprocessing. Before we go, if you're struggling with cell culture media challenges in your own work, I've got something special for you. Book a free consultation to help you get started with rational media design, leveraging automation and AI, and a lot more. If this is of interest, book a free call with me through the link in our show notes. Until next time, thank you for joining us. This is David Brühlmann signing off for today. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.